share some of my work in this event. My talk will be about microRNA as liquid biomarker for diagnosis and prognosis of breast cancer. I am Dr. Menha Swellam, Biochemistry Department. I'm the head for High Throughput Molecular and Genetic Lab, Center for Excellence and Advanced Science, Biotechnology Research Institute, National Research Center in Egypt. This work was funded by Science, Technology and Innovation Fund Authority through basic and applied research support grants, and the instrumentations needed for this work was funded also by capacity building grant. My talk will be about, this is some outlines of my talk, will be about microRNA definition, microRNA biogenesis and function, breast cancer epidemiology and incidence, then microRNA detection, and then I will show some of our lab works on microRNA and oncology. Okay, microRNA are non-coding RNAs found only in eukaryotes. They are small size with an average length of 22 nucleotides, transferred by RNA polymerase II from an independent genes. These microRNA play an important regulatory roles in both human, animal, and plant cells. MicroRNA biogenesis and function. MicroRNA biogenesis involves both nuclear and cytoplasmic processes. Pre-microRNA are transcribed by RNA polymerase II, and later processed by DORSHA to be microRNA. X14-5 transfer these pre-microRNAs from the nucleus to the cytoplasm, where DORSHA processed them into mature microRNA. These mature microRNAs can be selectively either incorporated with arginate, protein of RNA-induced silencing complex, and form MyRISC, then bind to their complementary messenger RNA, promoting their degradation or translational inhibit. Either the other pathway is as passenger strand, which is supposed to be degraded normally in the cytoplasm. MicroRNA either can be act as tumor suppressor genes or as oncogenes. Normally, the proper microRNA transcription, when they pass through the processes of transcription, processing, and binding to the complementary sequences on target messenger RNA, result in the repression of the target genes expression through blocked in protein translation or altered messenger RNA stability. So it results in normal rate of growth, proliferation, differentiation, and cell death. When these microRNA act as tumor suppressor genes, the reduction or deletion of a microRNA that functions as tumor suppressor lead to tumor formation. And this process carried out by a reduction or elimination of mature microRNA levels, which can occur because of the defect at any stage of microRNA biogenesis, and ultimately lead to inappropriate expression of the microRNA target oncoprotein, which give the purple squares, these purple squares. The overall outcome might involve increased proliferation, invasiveness, or angiogenesis, then decreased the level of apoptosis or undifferentiation or de-differentiated tissue, ultimately leads to tumor formation. So it results in tumor formation by increasing the proliferation, invasion, and angiogenesis, and decrease cell death. If these microRNA act as oncogenes, the amplification or the overexpression of microRNA that has an oncogenic role would also result in tumor formation in this situation, and this would be carried out by increase the amount of microRNA which might be produced at inappropriate time or in the wrong tissue, which would eliminate the expression of a microRNA target tumor suppressor gene and lead to cancer progression. So there will be increased level of mature microRNA occur because of the amplification of the microRNA gene and a consequently active promoter, increased efficiency in microRNA processing or increased stability of the microRNA, which is the open reading free. Our project was carried out on breast cancer, which is the second most prevalent cancer and one of the leading causes of cancer-related death among females in Egypt and worldwide. According to Globocan survey, about 2.1 million newly diagnosed female from breast cancer of the total cancer cases, with mortality rate nearly 6.6% worldwide. According to National Cancer Institute, breast cancer in Egypt accounts for about 38.8% of total malignancies among Egyptian females, and its incidence and mortality rates are among the highest in the Middle East. Late-stage diagnosis is common, and disease occurs at an earlier age than in Europe and North America. About 70% of breast cancer are sporadic, acquired during life, while 5 to 10% may be inheritable. Up to 90% of familial breast patients are due to mutations in BRCA1 and BRCA2 genes. The risk of breast cancer is based on clinical pathological features that primarily used for predicting metastasis at the tissue level. Some patients are difficult to treat due to disease heterogeneity and poor identification of metastatic disease. So early detection of breast cancer greatly improves the prognosis and treatment for patients. This is critical for optimizing the management strategies to this disease. CEA, carcinoembryonic antigen, and cancer antigen 15.3 are, and other circulating protein molecular markers have been widely used as non-invasive or minimally invasive biomarkers for monitoring patients with metastasis, but with insufficient sensitivities to diagnose primary breast cancer. Thus, a great necessity is highly needed to develop novel biomarkers for breast cancer detection with high sensitivity and specificity. So our aim in this study, which is one of the studies carried out from our project, is to detect the clinical impacts of panels of microRNA 21, 27, 155, 126, and 335, as minimally non-invasive liquid biopsy markers for early diagnosis of breast cancer patients. Then compare their expression with other, among those breast cancer patients with other benign breast lesions and healthy individuals, and then investigate their sensitivities, specificities versus the commonly used tumor markers in lab, CEA and CEA 15.3, then assess their prognostic efficacy as compared to other clinical prognostic criteria. In this study, we have recruited samples divided into 3 groups: breast cancer group, which are newly diagnosed breast cancer female before starting any treatment strategies, a group with benign breast lesion, and a healthy group which served as control group. Exclusion criteria were those patients that undergo mastectomy or a lumpectomy, or who received radio or chemotherapy, those with other types of malignancies or distant metastasis. The current study was approved by the Medical Ethical Committee from the National Research Center, and the recruited individuals were 200 primary breast cancer patients, 50 benign breast patients with benign breast lesion, and 40 healthy group who served as control. They were of age-matched Egyptian females. The experiments were carried out as detection the expression level of microRNA using real-time PCR, detect tumor markers, hormonal receptor status were carried out, and all demographic and clinical data were obtained as age, menopausal status, pathological type, clinical staging, histological grading, hormonal receptor status as estrogen receptor, progesterone, HER2. Analysis were carried out by using SPSS, and the fold change of the investigated microRNA was calculated according to the equation of Levelli. The association between the clinical pathological marker and demographic factors with investigated microRNA was determined. To detect the best sensitivity and specificity, receiver operating characteristic curve was plotted between the investigated groups for the detection of the efficacy of the investigated microRNA versus survival outcome. Both disease-free survival and overall survival were carried out. Disease-free survival was reported as the time from the first receiving neoadjuvant treatment strategy to distal or regional or local recurrence, secondary primary cancer, or contralateral breast cancer. Overall survival was measured from the date of the first diagnosis to the date of last follow-up or death of the patient. Of course, both were analyzed using a Kaplan-Meier statistical method and compared by log rank test. This curve shows receiver operating characteristic curve for investigated tumor markers CEA, C15-3, and 3 microRNAs, 21, 27, and 155. And as we can see from this curve, that area under the curve, which should be up to 1, and if it is below 5, so this means that this cutoff point cannot be considered. Of course, the best cutoff point, it is the point that discriminates between malignant and non-malignant group. And as we can see from the area under the curve, that nearly the CA, CA53, and the other 3 microRNAs were of nearly the same or nearly close to each other. When also the other 2 microRNAs, 126 and 335, were considered, they also were in the same, nearly the same values. Then we carried out comparison between the 3 studied groups: breast cancer, benign patient with benign breast lesion, and healthy control. And we reported that for CA and CA15-3 has sensitivity of positivity values were about 50% or 52% for CA and CA15-3, while that for other microRNAs it was higher. And as for 21 microRNA 21, it was 63.5%. 92% for microRNA 27, and 95.8% for 155, and for 126 it was 89, and for microRNA 335 it was 98%. So their positivity rate in breast cancer, newly diagnosed breast cancer patients, were higher than those of the routinely used tumor markers. When we compared the relation between the investigated microRNA and clinical pathological factors, both CA and CA15.3 did not report any significant with clinical pathological factors or demographic data, while for each microRNA investigated reported significant relation with, for example, clinical stage, as microRNA 21 was higher in late clinical stage as compared to early one, was higher with high-grade tumors. infiltrating duct carcinoma, patients with a positive lymph node. MicroRNA 27 was significantly elevated with patient with late clinical stages, higher grades, negative estrogen receptor and HER2 receptor. MicroRNA 155 was significantly increased with late clinical stages and high-grade breast cancer patients. MicroRNA 126 was significantly also increased with late clinical stages, high grades, and investigated invasive duct carcinoma and patients with positive lymph nodes. MicroRNA 335 was significantly increased in high-grade tumor. Then we detect the overall sensitivities, specificities, positive predictive values, negative predictive values for these investigated microRNAs versus the two tumor markers, CEA and CEA 15.3. As you can see from this table, the detection for breast cancer, the sensitivity for CEA and CEA 15.3 was 94%, while that for CA 15.3 was 52.1%. When we compare these values regarding other microRNAs, the remaining microRNAs or all the other microRNAs were exceeding these values, as that for microRNA 21 or microRNA 27, microRNA 126, and microRNA 155, microRNA 335. Interestingly, also, the detection for early stage breast cancer among the microRNA 335 and 155 and 126 and 27 were exceeding those tumor markers as well, and also the detection of low-grade breast cancer tumors, which as these two groups, those of early stage and low-grade breast cancer patients, considered patients with high risk. So it was our concern to compare the microRNA expression level among low-grade tumors versus those for CEA and CEA 15.3. And as we can see from this table, that the last four microRNAs were superior for that of CEA and CEA 15.3. Also, we were interested to carry out the expression level for these microRNA its relation with survival outcome. So patients, it was reported in this study that patients with either high expression level of microRNA 21, microRNA 155, or low expression of 100 microRNA 126 reported worse disease-free survival. For overall survival, a significant difference was reported between microRNA 21 and microRNA 126, as increased level of these of microRNA 21 and decreased expression of 126 reported worse overall survival. Also, for the relation between microRNA 335 was significantly related with worse disease-free survival and overall survival. So, we concluded that circulating microRNA expression level is a promising minimally invasive marker for diagnosis of breast cancer. Investigated microRNAs were superior over tumor markers for the detection of early stage and low-grade breast cancer patients. And that assessment of microRNA in blood facilitates their role as a potential prognostic molecular marker.
Hello, Dr. Menha. Sorry. Yeah, so sorry for the interruption, but we are running out of time. Can you please
I'm
conclude the
talking
presentation?
slide. Okay, this study was, as I told before, it was funded by the STDF, and we have the group, a member of our lab, have several publications regarding the role of microRNA in breast cancer, and then we shared for other types of cancer like acute lymphoblastic leukemia, glioblastoma multiforme, and we found that all of these microRNA can be used successfully as minimally non-invasive liquid biopsy markers for diagnosis and also prediction, the prognosis of cancer. Thank you very much, and thank you for listening.